Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. As you guys know from watching previous videos, these videos are intended for students who are enrolled in my class. I want them to learn the material the way that I present it. If you're in another instructor's class, then you need to learn all the details and all the stuff that they cover. It may be slightly different than how I do it. I hope these videos help all of you understand the material. If they do, please hit like and subscribe so that I can continue to get some feedback and continue to do new videos, better videos, and edit them and make them much more slick and more professional. But for now, I'm in a time crunch due to this coronavirus shutdown. Now, if you are in my class, we're doing a continuation of our bone lectures. We've done the formation, we've done an introduction to bone, we've drawn long bones and talked about the different shapes of bones. We did the bone wedge model with osteons and concentric lamellae and interstitial lamellae and all of that. And then in the last video I did what's called endochondral ossification, bone formation of most of your long bones of the body and how they grow within a hyaline cartilage model. We're going to move on to page 43 and we're going to talk about intramembranous ossification. The intramembranous means within a membrane, and if you remember, your skin is a membrane. It's called the cutaneous membrane, and it has two layers, an epidermis and a dermis. The dermis of your skin is where flat bones or dermal bones grow by a process called intramembranous ossification. Now, when I first typed this up, it's on the top of page 43, I have some steps here. And it followed our textbook, but we're getting a new edition of the textbook, and I don't like the way that I really typed this up. It's a little confusing, so we're not going to follow this exactly. I'm going to rewrite some details here of what's happening and draw this out for you so you can see what's called intramembranous ossification. So, intramembranous ossification is formation of bone within mesenchymal or embryonic tissue in the layer of your skin called the dermis. And we call the bones that form this way dermal bones or flat bones. The flat bones of the skull, what we call the calvaria or the cranium of the skull, your collarbone, your clavicle, the sternum, your ribs, all of the flat bones develop this way, even parts of the scapula, okay? So in intramembranous ossification, if you remember from early on in the semester, we drew out the layers of the skin. And if you think about it, the top layer of our skin up here, and imagine if I did this in a three-dimensional cutout, like a big flat piece of skin, okay? Well, where this corner is, I would still have this going back to this corner. So we're trying to look at this sort of a 3D cutout. And then down here would be all the the dermis, okay? So we have epidermis, and then we have the dermis of our skin, and then down here we would have hypodermis. Remember, the epidermis has a lot of areolar connective tissue and dense irregular tissue mixed in. And there's a lot of protein fibers here. And within this embryonic layer, this is occurring while we're in the womb. Within this embryonic tissue, some cells are going to differentiate. And the cells that differentiate are called osteoprogenitors. There's some cells called osteoprogenitors. We've talked about this. These are cells before they're making bone. Will mutate essentially or develop into osteoblasts. And as these cells generate into osteoblasts, they're going to start laying down some bone tissue along all these little fibers, these protein fibers, in all the different directions that they grow. And if you look at the edge, it looks like little pointy things sticking out. And they call those little points in here spicules, which means little spikes. So, within the dermis of our skin, some mesenchymal cells called osteoprogenitors will turn into or form into osteoblasts and start to lay down some little spicules of bone. And this type of tissue is called osteoid. Osteoid means close to bone or similar to bone or like bone, but it's not complete bone yet. Okay? And what they do is they simply, in order to form these spicules, they lay down calcium salts. onto the protein fibers that are already there. Now, over time, we will continue this growth. And essentially what this whole thing is a primary ossification center. So there's going to be a long three-dimensional band of these little spikes or spicules that grow through this flat layer of the dermis. Now, over time, it will grow out, and those little spikes or spicules, if I were to just focus on this tissue, will continue to grow out along stress lines and things. And what we are forming essentially is the trabeculae of bone. We've talked about what trabeculae are. They're plates of bone, and we grow these trabeculae out, forming this long plate of spongy bone. Over time, we're going to start to remodel some of this tissue here, and we'll lay down a thin plate of compact bone. And it will grow sort of in 3 dimensions. So we get a big flat piece of bone forming, and in the middle is a whole bunch of spongy bone with a little, and it's trabeculae, with some compact bone all around the edges. Now I know that doesn't exactly follow the steps in the note set. And if we wanted to follow the steps in the note set, within a layer of embryonic tissue, some stem cells, that's the first blank, called osteoprogenitors, differentiate into osteoblasts and begin forming calcium salts, and it forms bone. And even though there's a single blank there, essentially we're forming an ossification center or a primary ossification center. The osteoblasts start to get trapped in the matrix and become osteocytes. And then the cells from this ossification center will grow out in little spicules or trabeculae. And basically, the next line, step 3, would say small struts develop near the spicules and form the ossification center as it continues to grow. And those struts would be the trabeculae, okay? And then eventually a plate of spongy bone is formed, and we remodel and form compact bone around the edges of that, okay? And that's what a flat bone would look like. If you ever slice one of the skull bones open, it's got a thin layer of compact bone, and it's filled with spongy bone. We don't form a marrow cavity. Now, in the marrow cavity of long bones, we have yellow marrow. But in these spaces between the trabeculae of flat bones, we have what's called red marrow, red bone marrow. And that's where a lot of blood cell production occurs. So this doesn't exactly follow the steps. I'm actually going to put a link to a video that talks to this in my Canvas page for those of you who are enrolled in my class. You can do a Google search for these videos of the formation of intramembranous ossification, and you can find videos of it. But essentially, I need you to know these things. Intramembranous ossification is the formation of bone within a membrane of embryonic tissue. It is the dermis of your skin. And some stem cells called osteoprogenitors form or differentiate into osteoblasts and lay down a thin layer of spicules by depositing calcium salts onto the proteins, forming some osteoid. And then we start to grow those little spicules out, and as they grow longer, they're forming trabeculae of spongy bone. And then we remodel some of the tissue and put a layer of compact bone around that. That's pretty much it. So there's not a lot of put the steps in order, but I think you should know where it occurs in the body, know that it's the flat bones of our body, and know that we don't form a marrow cavity, but we fill all the empty space in here with red bone marrow. Okay. Now, that's rather short and sweet compared to all the steps of endochondral ossification. I'm a little bit more interested in endochondral ossification than I am intramembranous. Okay. Now, what we need to talk about is bone growth in length and bone growth in diameter for our long bones especially. Once our bones are formed, they continue to grow. And as they continue to grow, they're going to grow in both length and diameter. When we're born, our bones are little tiny things. The reason you're as tall as you are now is because your bones continue to grow longer. And that bone growth and length is called interstitial growth. I'm going to write all this out. And if they only grew longer, then our bones would be really long, skinny things like this meter stick. I knew there was one in this room somewhere. I saw it earlier. And if I try to put a whole bunch of weight on this meter stick on the ends, it's going to flex over time because it's not strong enough to withstand all the weight, and it would start to flex. I know you probably can't see that, but if I set this here and press down on it, you can see it starts to flex a little. Well, then our bones would be all bowed, especially our legs, from supporting our body weight. But if I made this really thick like a 2x4, I could stand on top of it and it wouldn't flex. So our bones need to grow in diameter as well as grow in length. Bone growth and length is called interstitial growth. Bone growth and diameter is called appositional growth. And we're going to write all these steps out. Why? Because I want you to learn it. Two, I want you to, or I need to have enough stuff to make a lot of test questions. So, I'm just kidding. So interstitial growth, this is how bones grow longer. Now, if you recall, when we looked at a bone during development, we have the marrow cavity, we have the diaphysis, we have the two epiphyses. I'm not going to put the spongy bone in here, but separating the diaphyses from the epiphyses is a plate of cartilage that we call the metaphysis or growth plate. And the reason it's called the growth plate is because this is where the bones are going to continue to grow longer. So you have to remember, and then we also have some hyaline cartilage out here. I'm going to leave the articular cartilage off. But that is hyaline cartilage that fills our growth plate. And we would have our secondary ossification centers here, and this was where our primary ossification center formed the diaphysis. Now, each edge of the growth plate is called a name. So right here on this edge of the growth plate, we call that the diaphyseal edge or the diaphyseal side. You'll see it in some books, they say the diaphyseal edge or the diaphyseal side of the growth plate, which would be the side touching the diaphysis. The opposite edge of the growth plate is called the epiphyseal edge or the epiphyseal side of the growth plate because it's touching the epiphysis. So when we go to write these steps out, essentially, here's what you need to know. On the epiphyseal edge of the growth plate, or on the epiphyseal side of the growth plate, I'm just going to give you the words that are in the blanks of my note set. For those of you that aren't in my class, I'll say all of this. On the epiphyseal side of the growth plate, some cells called chondrocytes, or we could say chondroblasts and chondrocytes, will continue to lay down cartilage. They're dividing by mitosis, and they continue to add cartilage to the process. Now, as they're dividing by mitosis and adding cartilage, simply what's going to be happening here is I'm going to add another line of cartilage and another line of cartilage and another line of cartilage and continue adding cartilage here and here. We do not remove the bone. What's happening is this. I'm pushing this epiphysis out further so that now the edge is not here, the edge is here. And the same thing would happen here. I'm pushing the epiphysis out. For example, if I put this chair here and I'm laying down cartilage behind me, I don't remove the chair, I'm pushing it behind me. So we're pushing the epiphyses out away from the diaphysis. And that's causing the bone to start to lengthen. The problem is this cartilage here would be easily fractured or ripped. So at the same time that we're adding cartilage on the epiphyseal side, some cells called osteoclasts and osteoblasts are going to remove cartilage and replace it with bone. And we will grow the bone tissue out further and add to the marrow cavity. And we do the same thing at the other growth plate. We're going to add some bone here and extend the marrow cavity so that now the diaphysis is keeping up with the growth. And the way we would write this out is these new cells are going to lay down cartilage. I just rewrote this this semester, so I want to make sure I put it the way that it's in your note set. And what that's going to do is push the epiphyses out. I changed the way that I'm teaching this because our textbook changed it a little bit, and I want to make sure that what I present goes along with our textbook so that students can study this more easily. It pushes the epiphyses out. Now in the third step, it says, at the same time, on the diaphyseal edge, these are just the words that are the blanks in my notes, osteoclasts will remove some cartilage, and osteoblasts will replace it with bone. Let me make sure that's how I have my... Yes. So essentially, osteoclasts are eating away the old cartilage, and osteoblasts are filling in bone behind it. And as we do this, the bone grows longer, but the growth plates don't get any thicker. They remain relatively constant in their thickness. Okay? Now, this bone growth will continue for quite a long time until we hit puberty, and our sex hormones will rapidly increase the bone formation much faster than they increase the cartilage formation. So over time, essentially what would happen is this. Imagine if on this finger I'm laying down cartilage, growing the bone longer this way, and this finger is laying down bone behind it, so that the growth plate is moving as we add tissue, right? But if bone formation, if they're moving at the same pace, the gap between them, the growth plate stays a certain thickness. But over time, what if these cells, the osteoclasts and osteoblasts, increase their activity faster than the chondrocytes are working? Then as I'm growing the growth plate, eventually we will start to remove cartilage and replace it with bone faster than we're laying down the cartilage. And eventually the growth plates get replaced with bone tissue. They close, so to speak, and then we stop growing in length. We've reached our adult height. For example, I believe I've talked about this before, but I'll talk about it again. The fourth step is already filled in my note set. We continue to grow the bone out throughout most of our life until we hit late puberty, and we close the growth plates and fill them in with bone. But if I were to look at someone's distal radius when they're like, 10 or 12, and the distal ulna, it will look like this. You'll see the radial and ulnar styloid, but you'll see where the growth plate is on an x-ray. There will be a gap in the bone. That's where the cartilage is, because cartilage doesn't show up on x-ray very well. If I were to look at this same individual later on in, a few years later, say when they're 23, the bone would look like this. There would be no more open growth plate. That means that person stopped growing. And if your x-rays are good enough, sometimes you can see the faint shadow of a line across there that they now call the epiphyseal line. The appearance of the epiphyseal line, the closing of the growth plate, indicates that you've stopped growing in length. You're not going to get any taller. So those are the steps for interstitial growth. Interstitial means between tissues, and between the bone tissue and the cartilage tissue here, we're growing longer. Okay? Now, again, if I grew my bones longer but not in diameter, then they would start to flex and bow over time. So that leads us to talk about what we call appositional growth. So I'm going to erase all this, and we're going to talk about appositional growth, which is bone growth in diameter. Now, this requires us to do a cross section of a long bone. So if I have a long bone here, and let's say I have some open growth plates, I'm just going to draw a generic piece of bone here. But I also know that I have this marrow cavity. If I do a cross section through this, if I cut it open and looked down on top of it this way, I would see a thick ring of compact bone filled with all those little osteons. I would see some spongy bone right around the inner edge here. I would have my perichondrium and my, periosteum and endosteum here and here. And you have to remember from a previous lecture, the periosteum and the endosteum have two layers. The inner layer, which is touching the bone, is called the cellular layer. The outer layer, which is away from the bone, is called the fibrous layer. It's very fibrous and tough. But the cellular layer is filled with osteoprogenitors and osteoblasts and some other cells. And there's even some osteoclasts roaming around in here. So now, if we were going to follow along in the note set, it's going to tell you interstitial growth is the process by which bones, appositional growth. I'm on another page. We're on page 44 of my note set. Appositional growth is how bones grow in diameter. It occurs at the same time that interstitial growth is occurring so that our bones can withstand more weight. And essentially what's going to happen is that osteoclasts on the inside are going to continue to digest away some of this spongy bone. And as they digest away the spongy bone, they're going to increase the size of the marrow cavity. And as they do digest this away, as the marrow cavity increases, the amount of spongy bone on the edge becomes less and less and less. At the same time, what's going to happen is some osteoblasts are going to migrate out of the inner cellular layer, and they're going to lay down a long piece of circumferential lamellae. Those plates of bone that grow all the way around the circumference here. If I continue to add circumferential lamellae over time, then it's going to push the periosteum out. And if I continue to keep adding rings of bone out here, it looks like the shaft of the bone would get very, very thick. But again, at the same rate, we're digesting away some of this bone here and increasing the size of the marrow cavity. And essentially what happens is I end up with a ring of compact bone, and I still have my spongy bone here, and I have my periosteum and endosteum. And essentially the thickness of the bone will increase a little bit, but again, these two things are happening at the same time, and as I do this in all directions, the diameter of the bone has grown. When I first started, the diameter of the bone was about this far across. Now we've extended it out to here, and the bone has grown much larger in diameter to support more weight, spreading that weight out over a larger surface area. So essentially, we're increasing the size of the marrow cavity, but we're adding circumferential lamellae, keeping the thickness of the bone relatively constant. And the bone grows in diameter. So if we were to go over the steps here where all the blanks are, let's do this for those of you that are enrolled in my class. Osteoclasts will continue to digest away the lining of the marrow cavity, increasing its diameter. At the same rate, osteoblasts from the periosteum will migrate out and lay down another ring of bone. And this is going to form what we call circumferential lamellae. And as I add circumferential lamellae to the outside, and keep adding, but remove bone from the marrow cavity, again, the diameter of the bone continues to grow, the thickness of the bone remains relatively constant, it gets a little thicker, and this bone growth in both length and in diameter will continue until we reach our adult height. Okay? So, know that these osteoblasts and osteoclasts are always remodeling bone and growing bone and shrinking bone, depending on dietary, or depending on your diet, how well you're eating and feeding your body the nutrients that it needs. If you were in a third world country and starving to death, then your bone growth and development would not be normal. People don't grow as tall or as healthy if you're starving them, and so their skeleton becomes thinner and weaker and more frail, more susceptible to fractures and other diseases. Okay. Now, I want to cover a couple of other topics, but we're going to do this in another video. We're going to talk about nutrition in bones, hormones in bones, and how to keep a healthy, strong body. We're going to talk about some interesting concepts that affect our bodies, and now we can really directly start relating our anatomy and physiology to our body with our diet, with our nutrition, with exercising and things that we do that can impact the health of your skeleton and the health of other tissues of your body. So I hope you learned something about interstitial growth and appositional growth, growth in length and growth in diameter. And I hope you know a little bit about intramembranous ossification. It occurs within the dermis of the skin, where some mesenchymal or stem cells convert into osteoblasts, form some trabeculae of spongy bone, and then we remodel some compact bone around it, forming the flat bones of the body. Okay. Hope you had as much fun as I did. I hope you learned something, and I'll see you in the next lecture. Do this stuff until you can't stand it, do it till you understand it, and do it till you can teach it to someone else without looking at your notes. Thanks for watching.